My name is Erin Henry. I'm studying at Wageningen University in the Wildlife Ecology and Conservation Department. My PhD focuses on the effects of pesticide mixtures on non-target and beneficial arthropods. So there are a lot of arthropod species that are not the intended target of pesticides. However, they can still be affected. And they provide a lot of different ecosystem services that are important. So, for example, pollination and also pest control. So I did a lot of work on work package 4 in the terrestrial group. And what we did was we came up with 11 different pesticide mixtures based on the residues that were found in the 11 case study sites. So what I did was I took these mixtures and I did some eco-toxicological testing on a few different beneficial arthropods. The first one is Amblysseus swersky. This is a predatory mite. It is used as a biocontrol agent in a lot of greenhouses. So what I did was a contact toxicity test. So what we did was we took these leaf discs, we placed juvenile mites on there, and we applied the pesticides at the application rate from the case study site, half the application rate, and 2 times the application rate. And what I found was that the effect on survival really differed between which case study site mixture we used. So in some cases we saw that nearly all application rates led to significantly reduced survival. Others seem to have no significant effect, and others you see at the application rate 2 times up. I also studied the effects of these mixtures on honeybees, Apis mellifera. It's a managed bee species, important for pollination, also for honey production. So the first test I did was an acute oral toxicity test. So in this case, we captured bees in the colonies, and we took them to the lab and placed them in these arenas, these little boxes, and we fed them the different pesticide mixtures in sugar solution. And the results were a bit similar to the mites because we saw really differing effects based on which mixture we looked at. So for example, the Switzerland mixture was toxic at nearly all concentrations that we used. For most of the mixtures, it seems like when we look at the single pesticide toxicity data, that the mixture toxicity was driven by 1 or 2 insecticides in the mixture. But these are preliminary results, so in the future I want to take a closer look at that and see if there are maybe some synergistic or antagonistic effects that we're seeing. So lastly, I also did a couple of colony experiments. So we took one of the case study site mixtures, so in particular the Dutch mixture. We tested the effects on honeybee and bumblebee colonies. And we used the same method where we fed treated sugar solution to these colonies. So for the honeybee colonies, we saw that there were significantly fewer worker bees, also significantly less brood in all stages, so the eggs, the larvae, and also the pupae, the capped brood. And we also saw significantly less stored honey, which is a problem for the survival of the colony. But this was as expected with the dose that we gave the colony. With the honeybee colonies, you would expect that if you have reduced workers due to the toxicity of the mixture, that you would also have reduced honey and reduced brood because there are fewer workers to actually take care of this. However, when we did look at the amount of capped brood per worker bee and honey per worker bee, we saw that there was still significantly less of capped brood in the treated colonies. And also, not significantly, but there is a relationship that there are fewer honey cells per worker bee in the treated colonies. And this shows that perhaps with the capped brood that there may be some larval mortality also. So perhaps the larvae do not survive long enough to become capped brood. And also with the honey, it could be that it could mean that the foragers are less efficient in collecting nectar. So that's what this maybe points to. For the bumblebees, we did a similar experiment where we treated the colonies and compared them to control colonies, but we saw no significant differences in the size of the colony or the number of bees, the mass of bees, or the number of new bumblebee queens that were produced. So at first we thought maybe this is an issue with the methodology because sometimes the bumblebees were not feeding on the treatment. But when we looked closer into this, we realized that the treated colonies were feeding significantly less on the treated sugar solution than the controls were. The controls were given sugar in the same way, just without pesticides. So we believe that this could point to some sort of repellent effect of the bumblebees to these specific insecticides that were in the mixture. A lot of these results are preliminary. What I want to do in the future is compare, for example, our survival data to the survival data of the single components, which is in the literature, to check if there are synergistic effects of the mixtures. What I'm also working on right now and finishing up is evaluating the differences in the gut microbial community of honeybees. So I sampled some bees at the beginning and the end of the colony exposure. And now we're going to look at if there are differences in the relative abundance of different important groups.